The Applied Biosystems TaqMan SARS-CoV-2 Mutation Panel is a scalable solution for follow-up testing of positive samples that allows you to detect one or many SARS-CoV-2 mutations, all on your current real-time PCR instrument. Starting with isolated RNA from SARS-CoV-2 positive samples, our unique workflow combines our gold standard Applied Biosystems TaqMan SNP genotyping assays with a one-step RT-PCR reaction to detect whether there are known mutations present in your samples or not. In this video, we'll show you how this one-step RT-PCR reaction works and what you can expect your results to look like. You will start with RNA isolated from SARS-CoV-2 positive samples. During reverse transcription, the RNA is reverse transcribed to cDNA, and then PCR amplification of the cDNA template begins. The master mix contains a DNA polymerase, and the reaction also contains a forward primer, a reverse primer, a FAM-labeled Applied Biosystems TaqMan MGB probe, and a VIC-labeled TaqMan MGB probe. Next, the cDNA template is denatured. The probe anneals to the complementary sequence of the cDNA strand. As long as the probe is intact, the proximity of the reporter dye to the quencher results in suppression of the fluorescent signal. Next, the forward and reverse primers anneal to the complementary cDNA sequences. During polymerization, the DNA polymerase only cleaves probes that hybridize to a target sequence. Cleavage separates the reporter dye from the probe. The separation of the reporter dye from the quencher dye results in increased fluorescence. The increase in fluorescence occurs only if the target sequence is complementary to the probe and amplified during PCR. Because of this, nonspecific amplification is not detected, and the fluorescent signal clearly indicates which alleles are in your sample. The mutant allele is detected by FAM dye. The reference or wild-type allele is detected by VIC dye. After your experiment, you will analyze your results using our Applied Biosystems Design and Analysis Software 2.5 or later with the Genotyping Analysis module. Results are displayed as cluster plots, like the one shown here. Samples that carry the reference or wild-type allele will cluster along the x-axis, shown here in red. Samples that carry the mutant alleles will cluster along the y-axis, shown here in blue. This concludes our How It Works video. To learn more about the TaqMan SARS-CoV-2 Mutation Panel, visit thermofisher.com/mutation panel.